Check it out now, y'all.
nanoHUB-U online instruction.
Hello and welcome back to our fourth lecture in week 2 of nanoHUB-U's course, An Introduction to Bioelectricity. So in this second week, we're talking about the chemical basis of electrical signals. And in this fourth lecture, what I want to talk about is post-synaptic receptors. So we've talked about action potentials. We've talked about active conduction, passive conduction, all of this within the context of the axon and the dendrites and the soma of the presynaptic neuron. What we haven't really talked about is what happens in the postsynaptic neuron and how do you get that signal from the presynaptic neuron to the postsynaptic neuron. We've talked about ion channels, but we haven't talked about the role that ion channels play in translating those signals from cell to cell. So that's what we're going to really focus on in this lecture. And why don't we just dive right in? So there are two kinds of synapse, broadly speaking. We'll talk about the least common first. So there are electrical synapse, or gap junctions, in which case you have the presynaptic neuron. So the presynaptic neuron, if you recall, is the neuron that is doing the talking. The postsynaptic neuron is the neuron that is doing the listening. The axon is what we'll look at for the presynaptic neuron, and we'll be looking at a dendrite or a soma in the postsynaptic neuron, most commonly a dendrite. So when the presynaptic neuron's axon terminates in this funny shaped ball, which is called a terminal bouton, when it terminates in this structure, it joins with the postsynaptic neuron. It is possible that those two membranes can become fused together, so to speak, so that the ion channels in one, the presynaptic neuron, become bonded with the ion channels in the postsynaptic neuron. And these ion channels are not like the other sorts of ion channels that we've talked about. These ion channels are much larger. And the way that they work is that when the action potential arrives at the bottom of the presynaptic neuron, the ions, the charge carriers, flow directly from the presynaptic neuron into the postsynaptic neuron. So in this case, neurons behave a lot more like electrical circuits, where we have metal connections between one structure and another structure, and the current just flows. The electrons flow between them than they do like most neurons, which we'll talk about next. So if you have a presynaptic neuron and you see an action potential in that presynaptic neuron, most of that charge will flow directly through into the postsynaptic neuron. And in fact, that single action potential will often be sufficient to create enough of a depolarizing current in the postsynaptic neuron to then cause that postsynaptic neuron to fire directly. Whereas most synapses are not electrical, they're chemical. And in a chemical synapse, a single postsynaptic potential caused by one of these synapses is typically not enough to get a action potential to occur in the postsynaptic cell. So in this second type of synapse, the chemical synapse, the presynaptic neuron has what are called synaptic vesicles. And these vesicles contain discrete, quantifiable quantities of what are called neurotransmitter. And when the action potential flows down the axon and reaches the terminal, it leads to these vesicles releasing the neurotransmitter into this gap between cells, which is called the synaptic cleft. And that release of neurotransmitters causes ion channels in the postsynaptic cell to open up and that neuron to depolarize or hyperpolarize, depending on whether it's an excitatory or an inhibitory postsynaptic potential. Okay, let's look at this in a little more detail. So you have the presynaptic axon of the neuron that is myelinated, and inside it has these structures drawn here in purple, which are these microtubules. And these microtubules aid in carrying synaptic vesicles filled with neurotransmitter down into the terminal. Then you have a sequence of events that occur that lead to a postsynaptic potential. The first of these events is that the synaptic vesicles have moved into the terminal. And there's two different ways in which that happens, and it depends on the type of neurotransmitter that we're discussing, but we'll get to that in a minute. But the synaptic vesicles have moved into the terminal. At this point, an action potential arrives down through a mixture of active and passive flow and saltatory conduction. It arrives at the terminal, and when it does so, it depolarizes the terminal. When it depolarizes the terminal, it opens up the voltage-gated calcium ion channels in the terminal. that happens, step 4, the calcium flows in to the cell. When the calcium flows into the cell, the vesicles bond, carrying the neurotransmitter, bond with the cell membrane. And when they bond, they open up and they release their neurotransmitter into the synaptic cleft. When that neurotransmitter has been released into the synaptic cleft, it in turn flows and then bonds with the ion channels in the postsynaptic cell. When that happens, those ion channels open up and then sodium ions rush in, the cell depolarizes, and you've got yourself a postsynaptic potential, which then propagates away from the synapse and in all directions, but the only one we care about is that it propagates towards the soma, and in the soma, the axon hillock, and there it adds up with all the other postsynaptic potentials, and if the threshold voltage is exceeded, then a new action potential is generated, and that travels down to the axon of that postsynaptic cell. In the meantime, the neurotransmitters that were left in the synaptic cleft are released and they slowly diffuse. In some cases, they are reabsorbed into the terminal and then repackaged in vesicles. Those vesicles themselves are reabsorbed and either refilled locally and prepared for the next release of neurotransmitter, or reabsorbed and the next dose of neurotransmitter comes in vesicles from the main cell body, from the soma, down these microtubules. So that's the circle of events that occurs during an action potential. The fact that you have these chemical synapses, and that in some cases you have an electrical signal that comes from the presynaptic cell and is sent to the postsynaptic cell with this intermediary step of being converted to neurotransmitters, which then open ion channels and lead to depolarization of the postsynaptic cell, was not a known thing always. It was something that was proven, and it's something that can be demonstrated. And one example that is in your textbook about how that is demonstrated is that you can take a heart and you can stimulate the vagus nerve. The vagus nerve is one of the cranial nerves. Remember, we talked about those in the first lecture. There's 12 cranial nerves, and they originate in the brainstem typically. There's a few that come directly out of higher structures, but most of them come into the brainstem. The vagus nerve is one of them, and it innervates much of many of the organs inside your body. One of those organs is the heart. It doesn't set the heart rate. That's generated locally, but it adjusts the heart rate up or down. So if you stimulate the vagus nerve, you can lower the heart rate. Now what you can do is you can take one heart, you can take two hearts and put them in separate beakers, right? Frog hearts. Put one in each beaker. You can stimulate the vagus nerve in the one heart, and that will lower the heart rate in that heart. Then you can take the fluid, the heart, that first heart is soaking in, and move that fluid into the container holding the second heart. And without stimulating the second heart, you'll see that the heart rate in heart 2 will also lower. And that's proof that the signaling coming from the vagus nerve electrically to the heart is converted at some point to a chemical signal, and that we can capture that chemical signal, translate it to a different organ, and replicate the same result. So you stimulate the vagus nerve, and you observe that immediately the frequency of the heartbeats and the intensity of the heartbeats drop. You take that fluid, you inject it into the environment of the second heart, and you observe the same drop in intensity, with a time delay, of course, as you move the chemicals from one heart to the other. So that's proof that in many cases you have this, in fact, in most cases in the brain, you have this step where you go from electrical to chemical and then back to electrical. Okay, so when we talk about these chemicals, we're talking about neurotransmitters. And so what makes a neurotransmitter a neurotransmitter? If you have a presynaptic cell and you have a postsynaptic cell and an action potential is delivered, a neurotransmitter is a molecule. It can be as little as a single ion, a calcium ion, or it can be as much as a large molecule peptide. But it's a molecule that, when released into the cleft, will elicit a postsynaptic potential in the second cell of the synapse, in the postsynaptic cell. That's the definition. Within that, there are a large number of neurotransmitters and categories of neurotransmitters. Some neurotransmitters, most neurotransmitters, are small molecule neurotransmitters. And these neurotransmitters are originally created in the nucleus of the cell, packaged in vesicles, or not packaged in vesicles, and sent down the microtubules where they are packaged at the terminal for release pending the arrival of an action potential. Calcium ion channels open, calcium rushes in, the vesicle bonds to the membrane, releases the neurotransmitter, you get your postsynaptic potential, and then those neurotransmitters are recycled. They're reuptaken into the cell through a separate set of ion channels, and that reuptake then allows the terminal to repackage those neurotransmitters into vesicles for release at the next action potential. This process of reuptake is critical in our understanding of many drugs and how they work. So if you take, for example, cocaine, and we'll talk about drugs a little bit more in the next week. If you take, for example, cocaine does not act directly on the neurotransmitter or on the synapse. Cocaine acts by reducing the reuptake, by inhibiting the reuptake of the neurotransmitter dopamine in the brain. So when your neurons fire, some of your neurons release dopamine. When the dopaminergic neurons fire and that dopamine is released, the dopamine is reuptaken more slowly when you have cocaine in your system than otherwise, which means that the neurotransmitter hangs out longer, which means that you have more excitation of the postsynaptic neuron than you would otherwise have. So you have an elevated level of neuronal excitability, which leads to a series of subjective experiences associated with the drug and with accommodation and addiction of the neurons in question. So those reuptake channels are very, very important. In a separate class of neurotransmitters, large molecule neurotransmitters, neuropeptides, these neurotransmitters are synthesized exclusively in the nucleus. They are packaged in vesicles in the nucleus, and then they travel down the microtubules to the terminal, where they are released. At the same process, when action potential comes down, calcium rushes in, the vesicle bonds with the membrane, the neurotransmitter is released, but then that neurotransmitter diffuses and degrades. It is not reuptaken, it is not repackaged locally. In every case, it's refabricated in the soma and retransmitted down the tubules. These neurotransmitters tend to be more powerful, but they tend to be smaller in concentration because of the need for this long process. I mentioned in a previous lecture that in some cases, there's so much diversity in these neurons and in these ion channels and in these synapses that in some cases a neuron can release two different kinds of neurotransmitters under different circumstances. And this is how that works. So if you take a particular neuron and you have the presynaptic terminal carrying both small molecule neurotransmitters that are packaged locally into the vesicles and large molecule neurotransmitters that are packaged in the soma and travel down the microtubules, you have stimulation of that neuron, an action potential comes down, the calcium ion channels open, calcium rushes in, and the vesicles fuse. But how much calcium rushes in? The quantity of calcium that rushes into the neuron is proportional to the amount of stimulus arriving at that terminal. And as you increase the stimulus frequency, so as you increase the frequency of the action potentials arriving at that terminal, you're going to increase the concentration of calcium not just immediately at the inner wall of the membrane, which is where most of the small neurotransmitter vesicles are packaged, but deeper in, which is where the large molecule neurotransmitters are packaged. And that means that if you have a low frequency, it's going to be a localized effect of calcium, and the small molecule neurotransmitters will be released. But if you have a high frequency, you'll have a higher concentration of calcium throughout. So the concentration of calcium here is shown as a purple density. So you have more purple, more concentration of calcium throughout the cell, and that will lead to not just the small molecule neurotransmitters being released into the synaptic cleft, but the large molecule neurotransmitters being released into the environment surrounding the entire synapse, the entire terminal and postsynaptic cell. So you can have one neurotransmitter or two neurotransmitters depending on the frequency of the action potentials traveling down the neuron. These can have dramatically different or complementary effects. The process by which this binding happens of the vesicles to the membrane wall is regulated by calcium, and it's regulated by a series of proteins. And the cleaving of these proteins and the bonding of these proteins, which is not something we're going to get into in this course, is what leads to the vesicles bonding with the synaptic membrane, opening up and releasing neurotransmitter. The reason I'm bringing it up is because there are a series of toxins. So we talked earlier about how cocaine affects reuptake. There are other toxins that affect the binding of these vesicles and the release of neurotransmitter. Amongst those are botulinum toxin and tetanus toxin. So the role that the Botox plays in neurons is to prevent the binding of vesicles with the postsynaptic membrane and the release of neurotransmitter. So it's a poison that interferes directly with the transmission of electrical signals in the body. And in fact, what it does is it leads to paralyzing of the muscles of, for example, the face, which is something that people do for aesthetic reasons, which is a mystery. Why would you inject poison into your own face? But it's also used in other societies by hunters, for example, to paralyze animal prey. It's used by animals to paralyze other animals for consumption, and it's one of nature's most common poisons. Tetanus is another example of a toxin that acts in this way. Okay, let's get back to postsynaptic receptors. So looking at postsynaptic receptors, we talked in the last lecture about how there are two broad categories. You have voltage-gated ion channels and you have ligand-gated ion channels. Within the ligand-gated ion channels, we said that there were several different categories. There were ligand-gated ion channels that were gated by the presence of a neurotransmitter on the outside of the cell. There were ligand-gated ion channels that were gated by the presence of a molecule like calcium on the inside of the cell. And there was another category of ligand-gated ion channels, which were what we refer to as secondary messengers. So I want to talk about those now. So first of all, to recap, in a regular ligand-gated ion channel, you have a neurotransmitter that bonds with the outside of an ion channel. When it bonds with that ion channel, the ion channel opens. When it opens, ions can flow through. For example, sodium can flow through the ion channel, depolarizing the cell and leading to a depolarizing postsynaptic potential. Alternatively, you can have what's called a metabotropic receptor. So the previous kind was called an ionotropic receptor. Here you can have a metabotropic receptor. And the way that a metabotropic receptor works is, for example, in the case of G-protein coupled receptors, where you have not a channel, but a receptor on the outside of the cell, or on the transmembrane. That receptor receives a neurotransmitter. When it receives a neurotransmitter, it releases a number of G proteins inside the cell. Those G proteins diffuse away from the point of their release, and they bond with effector proteins, or they bond directly with ion channels. If they bond with effector proteins, the effector proteins can release a large number of intracellular messengers, which can bond with a large number of ion channels. And those ion channels then open up, but they're opened up from the inside of the cell, so it's intracellular messaging, and the ions themselves flow through, and you have that same depolarizing or hyperpolarizing effect. So you have a number of steps, and because you have a number of steps, you would expect that the velocity, the speed with which this neuron or this set of this part of the membrane would respond to a neurotransmitter would be slower than in the case of an ionotropic receptor, and you'd be right. This is a slower effect. But the flip side of that is that you have gain. So this is an example of biological gain. You have a single neurotransmitter, and instead of that single neurotransmitter opening up a single ion channel and depolarizing your cell, that single neurotransmitter is now releasing a number of G proteins, which are affecting a quantity of effector proteins, which are releasing a potentially large quantity of intracellular messengers, which then open up a whole host of ion channels, leading to a much larger shift in polarization of the cell. So you have a slower process that allows a small concentration of neurotransmitter to result in a large postsynaptic potential. So it's a mechanism by which you see biological gain. Understanding all this, we can revisit postsynaptic potentials. So if you have an excitatory postsynaptic potential shown here in yellow and it exceeds the threshold, you're going to see an action potential in white. If you have an inhibitory postsynaptic potential that does not hit the threshold, then you're not going to see an action potential. More interestingly, if you have an inhibitory postsynaptic potential that goes up but doesn't reach the threshold, we're still going to call that inhibitory. So you'd think that depolarizing the cell would always be excitatory, and I alluded to this in an earlier lecture, but in fact that's not the case. And the reason it's not the case is because if you have a sum of postsynaptic potentials, the total voltage at the axon hillock will be the average, the weighted average of those signals. Any signal that is less than the threshold is going to drag the average down below the threshold. Even if it's greater than the resting potential, it still brings the average below the threshold. It'll bring the average above the resting, but you don't care about that. You care about the threshold. If you bring the average below the threshold, you make the neuron less likely to fire. And that means that it's inhibitory. So you can look at a number of different combinations in which you have an excitatory postsynaptic potential, a single one, and you get no action potential. You might have two excitatory postsynaptic potentials, and they add together, and you get an action potential. You might have an inhibitory postsynaptic potential, and you get depolarization. You might have an inhibitory with hyperpolarization or depolarization, but you don't hit the threshold, you don't get an action potential, and you can have combinations of excitatory and inhibitory. It's only when you cross the threshold that you get this action potential. Thank you very much, and I will see you at the next lecture.